Okay, let's get going. So this is 4.6 on immunosenescence. So this is just the gradual deterioration of the immune system with natural aging. Just like every other physiological mechanism that we have going on inside us, we're not built to last forever. At some point, the efficiency, the productivity, it's going to wane. It's going to decrease. So what does this look like? What are the implications of this? All right, let's look into it. It's not a uniform decline, and everybody's different. Um, Women live longer than men, so there's a component to that. But they also have an increased incidence of autoimmunity too, so there's a weird balance to look into. It affects both arms of your immune system, and it obviously would increase susceptibility to infections, right? If you are immunocompromised, you can't defend against most. You can't defend against things that you would normally be able to defend against. Certain cancers, right? We've been referencing that a good bit this unit. CD8 T cells, right? They're great for intracellular threats. And that's usually viruses, but it can also be oncogenic cells, tumorogenic cells. If I don't have a good surveillance, good CD8 T cell populations, then I am increasing the likelihood that if a tumorogenic mutation occurs, I don't have a cell that's there to get rid of it. So increased cancer rates, increased autoimmune rates. We'll talk about that a bunch in the last unit, but now I do not have the correct, pardon me, I don't have efficient selection mechanisms at play. They are starting to miss, right? The enzymes and the genetic mutation that has to occur to make sure that my receptors are perfect, that whole mechanisms, all those mechanisms are starting to show faults. We're getting more mistakes. My systems aren't filtering things that react to me at an efficient rate anymore. And then finally, if I don't have a good detection mechanism at play, well, I can't pick up even weakened pathogens from vaccines. So if I'm trying to be inoculated and protected against something, well, I don't have a lot of naive adaptive cells in circulation anymore, so I might not put up a robust a vaccine response because I don't have anything to really detect the the inoculation. All right. So what are some hallmarks of immunosenescence? Big thing is dysregulated inflammation, or inflammaging is the fancy like pop word for it now. Chronic low-grade inflammation without an overt infection. So I don't have anything outright infecting me. I don't have strep. I don't have pneumonia. I don't have anything really going on right now. But just because of my accelerated age, I would be secreting, inflammatory cytokines, enzymes, and reactive oxygen species that all contribute to this inflammatory tone, despite me not being infected with anything. So we have reduced surveillance and responsiveness, very much a poor response to novel antigens, and a waning memory quality. Despite that cell type being the predominant one, we're still just getting old. Like, these cells aren't going to be able to produce as robust of a response. And we'll talk about why the novel antigens are so scary in just a moment here. And then impaired resolution of inflammation. I told you guys already, I'm already rebuilding this class, and I want to do a whole talk on just inflammation and the fact that we need to actively resolve that inflammation on every single immune response. We've talked a lot about this unit on how bad the immune response can be when the wheels fall off the wagon, when it just goes and goes and goes. There is an active effort to resolve inflammation after we believe the stimulus has been removed. Okay? If we can't do that, then we have these cytokines perpetuating. All right? So part of this directed resolution is there are such things as anti-inflammatory cytokines. But if those aren't being secreted correctly, if my aged old cellular mechanisms aren't working properly, then I don't have those anti-inflammatory compounds released, and inflammation persists. And failure to clean out dead cells and pathogens is a big thing too. If my macrophages aren't working as efficiently as they should be, right? This is where we go back to maybe increasing autoimmune rates. A big thing with poor debris clearing is lupus. That's one of the postulated etiologies of the disease, one of the thought causes. Lupus is an autoimmune disease that binds DNA. Well, your DNA should never be floating around. It's only floating around in cases of like neutrophil extracellular traps or any type of apoptosis in general. That's usually not a problem because it's cleaned up. Not if the macrophages are slacking. So you see, there's just a lot of things we take for granted that these cells do. Like, yes, if you've taken a couple biology classes, macrophages clean up, yada, yada. But if they don't do that, it's very bad. It is very, very bad and can lead to really terrible conditions. So at the innate level, remember the innate immunity, fast, non-specific, we start to decrease accuracy. So we're just sending these innate cells everywhere or where they might not need to be, or maybe they're releasing compounds when they shouldn't be, and they're less regulated. So again, those anti-inflammatory cytokines, maybe they're just not getting to where they need to be. We see a preserved count, though. We don't really see a drop in these numbers, and we'll speak about that in just a moment. Next up is adaptive immunity. Remember, targeted memory forming, this preserved immunity. We see a reduction in diversity, proliferation, and precision. Diversity because if you think about all these immune responses that we've been talking about, if you've lived to 80 plus, right, think about how many memory T cells and B cells you have ready and waiting, okay? But also think, well, that thymus is completely gone, and I'm not making maybe as many B cells as I did when I was a young buck, so all I really have on the adaptive front are my old heads, my scholars that have already fought and already produced a response. Now, great if I come across that antigen again, but what do I do if I come across a novel antigen? That's something to think about. And again, these proliferation mechanisms and just the fighting in general that these cells do is not as efficient due to accelerated age. And here's a cool little diagram I found that breaks down a lot of the arms of senescence, if you will. DNA damage, of course, just mutations over time. I forget the number. We'll talk about it next unit. I want to say like there's millions of mutations or of sequence breaks in DNA a day, if you will. So, yeah, a lot going on there. Usually, though, our reparation or our repair mechanisms work very well. It just takes a couple decades on the planet for those to start to wane on their efficiency. Mitochondrial dysfunction is a big thing too. That's related to oxidative stress, metabolic dysregulation. So if I have these reactive oxygen species floating around, well, that's going to really hurt my mitochondria. And remember, mitochondria, powerhouse of the cell. Well, what is getting power? Getting power you get from breaking chemical bonds. That is from metabolism. So metabolic dysregulation. So oxidative stress messes with my mitochondria, which messes with my metabolic regulation. So all of this really works together here. How my genes are packed or how my DNA is packed gets messed up. We'll talk about telomeres in a moment here. Maybe I have too many or too few chromosomes. Maybe replication just isn't going as efficiently as it should, or I'm not getting that separation of chromosomes that I usually get. And this is going to be a great talk in Unit 5 I'm really, really excited for. If all of this stuff is impaired, we do run the risk of oncogene activation. And that is one of the genes that when functioning normally, it is regulating cell cycle behaviors. So, the different phases of what a cell should be doing. If that gene gets mutated, well, then that cell is going to be perpetually replicating, and that's literally cancer. That's tumor. So, something to think about, but we'll talk about that more later. A big thing to consider is thymic involution. So when the thymus is replaced with adipose, fat, and connective tissue. Okay? This starts in adolescence, usually around puberty time, and is just constantly going on. You have exceedingly decreased amounts of functional thymic tissue as you age. And by this advanced age, by the elderly age, you functionally have no thymic tissue. So we give a huge decreased count in naive T cell numbers, right? Remember, this was T cell college. T cell college is gone. They can't do that. So I can't make nearly as many T cells or naive T cells as I once was able to. So that means we're really reliant on this memory response. I hope to goodness that any disease that I see, I've already fought, because that's really all that's where the best defense that I have is. So it's kind of backwards. You want to see the foes that you've already seen. Okay. So memory Ts dominate, causing us to rely on a narrowed repertoire. And this is that idea. Okay, we really don't want to see a naive antigen, right? Think to COVID. Okay, think about how detrimental that was to the elderly population. One of the reasons for that postulated can be, well, those naive T cell numbers are just not going to be nearly as high in that population of individuals. And coronaviruses were relatively rare. You might have come across one and produce maybe some cross defense against, but we saw that was extremely rare, and those populations suffered, unfortunately. So that naive T cell drop can have a lot of detrimental implications. Then we have a shift in hematopoiesis here. With age, hematopoietic stem cells show a skew towards the myeloid lineage. Well, think back to Unit 2. Think back to that diagram. What was in that myeloid lineage? Well, that was mostly my innate cells. So now I have more of an innate bias. I got an increased number of macrophages, and neutrophils. I don't know why I did that. I'm making myself laugh. And like basophils, eosinophils. So we skew towards innate cells rather than adaptive cells, thus perpetuating this decreased arm of my adaptive immunity. Okay? Or the decreased potency, rather, of my adaptive immunity. Another thing that occurs with age is just fibrosis, general laying down of collagen. It's just part of this aging process. And let's think about it at the organ level, right? A lot of these organs that we've talked about are filters to some degree. They're sieves. All of my lymph nodes, my kidneys, my spleen, my liver, they all act as sieves, filters. When I lay down a mesh network on top of my filter, well, it's not going to work nearly as well. So that's what's kind of happened here. We form collagenous connective tissue where I really just don't need it. And that impairs clear separation of the different zones in these secondary organs, these secondary lymphatic organs. Again, that fibrosis increases with age, and now the filters and the roads that I use to transmit and transfer these lymphocytes and white blood cells in general, well, now they're getting traffic jams. So I have stymied trafficking of these lymphocytes due to increased deposits of collagen and fibrin. All right. Another concept that's really, really important here, but we should definitely know, and this has implications well beyond immunology in this course in general. It's just a cool thing to know, and it's been being researched since I can remember. So we know that this is a problem, but what can we do about this? Well, first, let's talk about it. So telomeres are the protective caps at the ends of chromosomes. So everything highlighted in red here. And they act as a protective cap. So the genetic material at the end here is, from what I understand, genetic nonsense. It codes for nothing. It's not. It's inert, if you will. So it just forms as a buffer. That way, once I get to the actual, like the start of the book, that all of that text is maintained. All of those genes are safe and ready and willing to be read. During unfurling of this chromosome and reading of this DNA, we're going to eventually shave down that telomere. Each time that is read and replicated, I'm going to reach back into my genetic notes from undergrad, but you have those primers, those little chunks that are taken off. And eventually, that chunk is not going to be telomere. It's going to be actual DNA. And when that happens, or if the telomere just gets too short, maybe we don't get to actual DNA, but we just get to the nub end of the telomere, we just stop. We say, okay, we can't do it anymore. My telomeres are used up. And that's something we call replicative senescence, when the cell says, hey, I literally can't replicate anymore. So it just hangs out, which is also not good because the cell's not really meant to do that. And we'll see what a senescent cell ends up doing in just a moment. To the B cells, a similar arc to the T cells. Naive B cells from the marrow decrease because we have that innate bias during the as we age. Class switching and affinity maturation decrease in efficiency just as those natural mechanisms start to wane. Thus, germinal center formation decreases because I don't have naive B cells coming in to even start the formation of those. Okay? Right? A naive B cell would see its new antigen and then start to do this whole affinity maturation process. That's just not happening if I don't have naive B cells. So, what perpetuates? Low affinity, because I don't have affinity maturation, and potentially autoreactive antibodies, because my positive and negative selection mechanisms are starting to get old alongside me. So that is the consequence here. I don't have this going on, so I have less affinity, a poor handshake, if you will, between the antigen and the antibody, and potentially autoreactive antibodies, since my natural mechanisms of selection are starting to fade. Okay, and now a big take home here. I love this diagram. Oxidative stress kind of works like a rotting tomato. So you have normal cells. When you have too many free radicals, this is just really detrimental to the cell. Okay, so these molecular level mechanisms. Accumulation of these reactive oxygen species, ROS, which is just a free radical oxygen, an oxygen anion floating around. I've said it a bunch of times in class, but I'll say it again. Think of them like bullets at the molecular metabolic level. It damages DNA, proteins, and lipids. It can mess with cell cycle and mess with division in general. A big thing that I remember them affecting is mitochondrial efficiency. Okay, the molecular the reactions going on within the mitochondria, the Krebs cycle, oxidative phosphorylation, and plenty other chemical reactions that I'm forgetting right now, they're not going to benefit from an oxygen bullet coming through and disrupting equilibrium or disrupting the specific bonds that need to be made for those chemical reactions to occur successfully. So this completely just disrupts all of this stuff here. Usually we have that under control, right? Like I said, that resolution from inflammation is extremely important because of how damaging these can be, and our neutrophils secrete a lot of that. But in this case, if we have an inflammatory tone, if we have that inflammaging, then we have neutrophils everywhere that are potentially getting activated for no reason. We have more of them than we do adaptive cells due to the innate bias, and this just starts to compound. Another mechanism to consider is something called epigenetic drift, and this is DNA modifications that control gene expression without altering the sequence. So I'm not here like turning. You're not turning into a zombie as you age. You're not turning into a mutant fish or anything. The genes themselves are staying the same, but it's how they're packaged that might get uh, confused or difficult. So let's go back to this image because I'm just talking in circles right now. So this is just a fancy or a simple drawing of how the genes might be packaged. Okay. You have proteins that do this called histones, and really just how those are folded down determines what regions of the genome will be read. Will be read by the different cellular machinery. And remember, it all comes down to the genes and what genes are being read. So if the packaging is showing incorrect genes on top to be read, well, then you just get this haphazard activation and silencing of genes that maybe weren't supposed to be activated in the first place. So we're just kind of flipping on and off light switches as we go along. It's a bit more complicated than that, but if you just think about it as DNA is getting packaged a bit incorrectly, I think that'll get you home. And finally, what's the problem here? Well, these senescent cells, they're rather dormant and non-replicating, but they just tend to secrete these inflammatory signals, probably due to the fact that they're not used to persisting this long. That persisting beyond the cell cycle is something that's inherently bad, that's associated with cancers. So they're secreting these inflammatory cytokines, thus furthering this inflammatory tone that contributes to inflammation. Okay, so kind of a vicious cycle here. But yeah, that is everything I wanted to cover here. Oxidative stress and these reactive oxygen species are really the big drivers behind this. But this isn't the whole story, and I'm not the end-all, be-all professional on this by any means whatsoever. And there has been some cool research showing that some flu vaccines that's in the elderly populations, because they had all of these innate cells, or I'm sorry, over here, where was it? Yeah, because they had this innate bias and had all these cytokines and innate cells at the ready, they actually produce a pretty solid immune response. So I guess it just depends on the stimulus, and there's a lot of flux to this story that needs to be understood. But it is really interesting, and the more we learn about this, the better we can protect this population. So a novel antigen isn't so scary and dangerous, and hopefully that never has to happen again, but hopefully if it does, there are better defenses at play. All right, thank you guys very much, and I will see you soon. Okay, there we go.